What if it was possible to hand-select traits you want to see in your baby, or even prevent certain genetic diseases from being passed down? The process of genome editing allows us to generate what is known as designer babies. Today we will be outlining the background of so-called designer babies, the technologies required to make this happen, and the current developments of a computer chip that will make this process much more efficient. The creation of designer babies is a controversial process that involves genetic interventions by implementing an embryo that has modified DNA fragments designed to correct the disease-causing genetic errors. Some think that the creation of designer babies is like creating a Build-A-Bear, where scientists have the power to hand-select individual traits. Although this application is theoretically possible, in reality it is a much more complex process that hasn't been successfully executed yet. However, Adam Nash was the first baby born with an artificially edited genome, and thus called the first designer baby. Adam's older sister was born with a rare genetic disorder. In order to effectively treat this disease, she needed stem cell treatments from someone with a specific and disease-free genetic makeup. In the embryonic state, Adam's genome was profiled to ensure it would be a match for his older sister. One of these genome editing technologies is CRISPR-Cas9. CRISPR consists of two main components, the Cas9 protein, which cuts the DNA, and the guide RNA, which recognizes the DNA sequence that needs to be edited. The mechanism of CRISPR has been described in a 2019 review paper by Rodriguez et al. First, the guide RNA is engineered to identify the specific sequence in the genome that needs to be edited. This guide RNA is attached to the DNA-cutting Cas9 protein, and this complex enters cells. The guide RNA looks for the sequence needing to be edited and cuts DNA. Cas9 helps to edit the existing genome by either adding, deleting, or changing the sequence. You can think of this like using spell check on a Word document. Say there's a sentence that needs to be edited. She go to the store every Wednesday. You can think of the sentence like the DNA sequence. The guide RNA is like the program in your computer that searches for errors. Once it's found the error, she go. Cas9 is the feature that actually changes the error from she go to she goes. While this process is much more complex than using spell check on your paper for English class, the results are the same, changing the original material with the intention of making it better. Hypothetically, CRISPR should work similarly to fixing a recurring typo in the document with an autocorrect feature. However, this process is highly complex and oftentimes imperfect. CRISPR-Cas9 molecules may sometimes target the wrong genes, replacing healthy segments of DNA with those that do not belong there, like when your phone autocorrects a correct word to an incorrect word, resulting in a completely different meaning than intended. This is because it is difficult to target a specific 2 or 3 base pair sequence in a genome of 3 billion base pairs, like manually looking for the needle in a haystack. In a 2019 study by Jones Jr. et al., who are researchers at the University of Texas at Austin, are developing a sequencing chip called CHAMP, which stands for Chip Hybridized Affinity Mapping Platform, that uses machine learning to allow for more effective CRISPR gene editing. It works by analyzing all the possible sequences that the CRISPR protein would target and uses machine learning algorithms to find the most common sequencing with errors, like a Control-F feature. With CHAMP, it is possible for CRISPR to fix thousands of errors at once with exponentially less error, whereas beforehand you had to manually find the specific sequence error. As you know now, the opportunities and possibilities associated with new genome editing technology are almost endless. Genome editing via CRISPR-Cas9 is currently being explored mainly as a mechanism to cure genetic diseases, but it has the potential to be used for altering non-disease traits. This controversial application of gene editing is referred to as creating designer babies, and the ethics behind it are still being debated. The use of these gene technologies shows us that fascinating but potentially scary applications of genome editing are on the horizon.